Good morning. Here I am discussing about the topic enzymes involved in DNA replication. We know the fundamental property of living organism is their ability to reproduce. Organisms produce offsprings that are similar to themselves. The process occurs by which a single parental cell divides to produce two identical daughter cells. And in this process, genetic material must be duplicated prior to cell division. So the daughter cells receive a full complement of genetic information for the progeny. We know DNA, the genetic material, that is able to perform two processes. First one is autocatalysis and the second one is heterocatalysis. In autocatalysis, DNA would make a DNA molecule and the process is known as replication, and in heterocatalysis, DNA would make an RNA molecule and the process is known as transcription. Autocatalytic function of DNA, that is a multi-step process, and a number of enzymes are involved in this reaction. We know DNA is a helical molecule, that is the two strands of DNA coiled around each other. So, if you want to replicate the DNA molecule, first we want to reopen the strands. So, the two strands get separated and both we can get the new DNA to be synthesized. So, replication means that is a synthesis of daughter nucleic acids molecules identical to the parental nucleic acids. This accurate and complete replication of the DNA is essential for an organism to reproduce. It is a basic biological phenomenon, it is a basis of biological inheritance. DNA is made up of double helix of two complementary strands, and there are many enzymes, they participate in the process of replication. So, come to the topic, the enzymes involved in DNA replication. This is a list of enzymes involved in the process of DNA replication. They are DNA helicase, topoisomerase, ligase, single strand stabilizing proteins or SSB proteins, primase, DNA polymerases and phosphorylase. In coming to the first enzyme, DNA helicase, the DNA helicase is discovered in E. coli in 1976, and they are the class of enzymes vital to all living organisms. DNA helicase is also known as helix destabilizing enzyme. Function of helicase is to open the strands or unwound the coil or helix. This happens by breaking the hydrogen bonds between the two strands. Then the two strands being separated. If you imagine a rope that is made up of 2 or 3 strands, separating these 2 or 3 strands, that is what we needed. This is double-stranded DNA molecule. This is a helicase enzyme. The helicase enzyme acts on this double-stranded molecule and the strands become separated and become single-stranded. So, the function of helicase is to unwound the DNA by breaking the hydrogen bonds. Then come to the second enzyme, that is topoisomerase. Every cell has enzymes that increase or decrease the extent of DNA unwinding. They are called topoisomerases. Topoisomerase is also known as DNA gyrase, and that act on the topology of the DNA. So, it is known as topoisomerases. Using helicase, the hydrogen bonds between these two helix had been broken, that is by the two strands separated. If, as we said, we compare it with a rope, suppose we pull these coiled ropes apart, these get separated, and because of the tension formed in the strands, they undergo recoiling. So, what is the job of topoisomerase is, topoisomerase release the tension. What is the main role of topoisomerase is release the tension or remove the tension from the individual strands. Topoisomerase binds to the double-stranded DNA and cuts the phosphate backbone of either one or both of the DNA strands. Topoisomerase is an enzyme that can change the linking number. Linking number is, that is a topological property of DNA molecules. It can be defined as the number of times the second strand pierces the second strand surface, that is linking number. There are two classes of topoisomerases, topoisomerase 1 and topoisomerase 2. Type 1 topoisomerase and type 2 topoisomerase. Then come to the second enzyme, that is DNA ligase. The main action is that is the ligating enzyme. DNA ligase is a specific type of enzyme that facilitates the joining of DNA strands together by catalyzing the formation of phosphodiester bonds. When topoisomerase cuts the DNA helix for removing tension, the cut pieces then get attached with the help of enzyme ligase. Later, when we come to the actual process of DNA replication, ligase, the pieces of DNA forms and they attached helped with the ends of ligase. It's an ATP required reaction. Then the next enzyme, that is single strand binding proteins or SSB proteins. After helicase has opened the strands, then topoisomerase cuts, rotates and reseals. Then SSB proteins, they make them the single stranded DNA stable. The SSB protein prevent the re-annealing because the strand has the tendency to anneal. So SSB proteins prevent the re-annealing, protect single stranded DNA from being digested by other nucleases and prevent secondary structure formation, thereby allowing other enzymes to function effectively on the single strand. This is single strand binding proteins. By the action of helicase, double stranded DNA unwound, and in this single stranded DNA, single strand binding proteins assemble, and this single stranded binding proteins prevent the re-annealing, rejoining of the separated strands. Then come to the next enzyme, primase. DNA primase is an enzyme that involved in the replication of DNA and is a type of DNA polymerase. Primase catalyzes the synthesis of short RNA segments. Primase is an enzyme. Primase catalyzes the synthesis of short RNA segments called a primer that is complementary to the single-stranded DNA template. This primer is actually a DNA-dependent RNA polymerase. This primase enzyme synthesis RNA primers in the 5' direction. This is a primase enzyme. Primase enzyme synthesis RNA primers at the 5' end. Then the next enzyme, that is the polymerase. DNA polymerase then helps the removal of primer because this primer is an RNA molecule. So we need to remove this primer. So DNA polymerase 1 helps the removal of primer and filling the gaps. In the next enzyme, DNA polymerase 3, that is mainly involved in prokaryotic DNA replication. DNA polymerase are the most important enzymes that synthesis DNA molecules. All other enzymes are helping enzymes. The functioning, it's the DNA replication process actually done with the help of DNA polymerase. All other enzymes are helping enzymes, so in which it is actually participating in the process of DNA replication, that is DNA polymerase. Also perform proofreading and error correction. This is a large molecule. This DNA polymerase is a large 3 is a large molecule. It has 3 subunits, alpha, epsilon and beta 2. Each subunit has its own functions. The alpha subunit, that is a catalytic unit that helps the formation of bonds between the nucleotides, the incoming nucleotides. Then the epsilon subunit, it has a proofreading function. If any wrong nucleotides are added to this, the subunit proofreads it and DNA polymerase ultimately going to check and replace that faulty nucleotides with the correct ones. Then beta 2, it acts as a sliding clamp-like subunit. This DNA polymerase can hold on to that separated strands and it can replicate. An interesting thing about DNA polymerase 3 is it can replicate both the directions simultaneously, so can act as a dimer. DNA polymerase can act as a dimer. This is the action of DNA polymerase enzyme. It's a double-stranded DNA arranged in chromosomes in the nucleosome structure. Then the double-stranded DNA, helicase enzyme unwind the double-stranded DNA. Then formation of replication fork occurs, and the DNA polymerase in 5' to 3' direction, new DNA molecules are synthesized in leading strand and lagging strand. This is a family of DNA polymerases in prokaryotes and eukaryotes. In prokaryotes, there are 5 different types of DNA polymerases: DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, DNA polymerase 4, and DNA polymerase 5. But in eukaryotes, more than 15 polymerases are discovered, but only 7 with the non-functions. DNA polymerase alpha, DNA polymerase delta, epsilon, and DNA polymerase gamma are the important ones. These are the prokaryotic DNA polymerases with a function. The prokaryotic DNA polymerases mainly 3 types: polymerase 1, 2, and 3. Polymerase 1, that is its function is removal of primer, and DNA repair and removal of DNA primers. It has an exonuclease activity at 5' to 3' direction and 3' to 5' direction. In polymerase 2, it has a DNA repair function and exonuclease activity only at 3' to 5' direction. In polymerase 3, its main enzyme of replication. DNA polymerase 3 is the main enzyme involved in prokaryotic DNA replication, proofreading and editing functions also, and exonuclease activity in 3' to 5' direction. This is the eukaryotic DNA polymerases. At least 7 polymerases with non-functions in eukaryotic DNA polymerases. The types are DNA polymerase alpha, DNA polymerase beta, gamma, delta, and epsilon. DNA polymerase delta is a major replicative enzyme in eukaryotic DNA replication. DNA polymerase alpha, its polymerase activity, that is the replication and DNA repair functions. It has no exonuclease activity. And DNA polymerase beta, it has DNA repair function and no exonuclease activity. Gamma, it has replication in mitochondria, mitochondrial DNA replication, that is involved in DNA polymerase gamma is involved in mitochondrial DNA replication and exonuclease activity at 3' to 5' direction. And DNA polymerase delta, it has replicative functions and DNA repair functions and exonuclease activity at 3' to 5' direction. And DNA polymerase epsilon is only replicating function and exonuclease activity at 5' to 3' direction. Then coming to the last enzyme, that is phospholase, helps the addition of phosphate groups. Phospholase helps the addition of phosphate groups. When nucleotides are added, they need to be activated. Those are all monophosphates. They are added in the form of monophosphates. They have to be turned to triphosphates. So phospholase helps the activation of phosphate molecules. So this is the entire process takes place in the DNA replication, role of enzymes. The first enzyme, helicase, helps the unwinding. Helicase unwinds the DNA and the formation of replication fork occurs. Then topoisomerase release the tension formed due to the unwinding of DNA strands. Then primase initiates the synthesis of RNA primers and DNA polymerase synthesis complementary DNA strands. Then after primer removal, the gaps were joined, or that is the gaps were filled with the help of enzyme DNA ligase. SSB protein binds to stabilize the strands and prevent the rejoining function. That's all.